Uh, okay, this is being recorded, I think. It's giving me options to pause recording, so I'm guessing it's being recorded. Um, so yes, we want to cross over, right? So a double-stranded DNA break is literally doing what my eraser is doing here. It's doing this, right? And my eraser is literally just not working on this chromosome. My eraser is literally making breaks in there so that I can substitute it for a different color. And this like the whole thing gets cut off. Like it's literally like an eraser making a hole in it so you can kind of patch in something else. Does that make sense? Okay, cool. Um, and remember, this is a chromatid. So this right here, this is a chromatid. Going all the way down, this line is a chromatid. A chromatid is a line of chromatin. So this already has two strands of DNA in it. So this is two strands of DNA. This is two strands of DNA. That kind of stuff, right? Um, okay. And I also got a question. How would you possibly word a question like this? I could imagine a like a true false, and it would be like, uh, no, she wouldn't do a true false. She'd do a four question MCQ. And it would be like, oh, chromatin, uh, or chromatids are held together by, and then it's like, uh, A, synaptonemal complex, B, cohesin, C, centromere, and then D, topoisomerase. So that should like scare you, like, oh, what the hell is topoisomerase? Um, so like it'll be something like this. That's what I imagine. Or she'll have a long ass paragraph of a question where the only word, and we'll see this in the practice final, where the only word you have to pull out is chromatids. Like she'll say like, oh, this is responsible for this and responsible for that, and she'll have like a whole paragraph of like names and weird protein names that she's like trying to throw you off with, and the only important word in there is chromatids. We'll see this example of this question. Um, what do I mean by 2x? So these are lines of chromatin, and chromatin is this. So these are histones, and this is DNA, and DNA is already double stranded. So histones hold together double stranded DNA, right? DNA looks like this already. It has two strands. And so that's why a double stranded break is like that. Because you're already breaking two strands of DNA. And then I'm saying this is two strands of DNA, this is two strands of DNA. Right? Like you're only breaking one of these pieces. See that? But it is called a double stranded break because inside here is actually like that, essentially. Does that make sense? Okay. Let's go ahead and so this entire thing, so all these names, whenever you see names like this is important. SPO 11 is important. That's about it. Like the entire process of crossing over, I don't think is that important. It's kind of, hey, this is what happens, this is what does the thing. And it's like, you'll actually see an example of the question on this on the practice final that we cover. Um, they do just bring up like functions of proteins, and it's not this deep. So here you see homologous chromosomes being held together by the synaptonemal complex. So this picture is essentially just this, and then oh, what happened? Oh, okay, that's weird. Okay, no, I'm just trying to. Anyway, yeah, what I was saying. Why can I not annotate? Oh, nor what happened? Okay. This is this essentially, and then the synaptic complex would look like this. It's literally like this. So that's this part right here. These lines right here. That's that. Does that make sense? Um, okay. And SPO11 here is important. SPO11 is the only protein here that's really that important, and it's because it does what? Somebody have an answer for me. Yes, absolutely. Um, it not quite. It's so Sergio got it right. It breaks well, it forms breaks. So break formation is right. It forms those double stranded breaks. So SPO11 is my eraser going like this. SPO11 does that. And so these will exchange with each other and yada yada. I can only draw this so many times before I start messing up my art, as you can see. There you go. And so SPO11 is the eraser that does that. Cool. Okay. And then moving slightly down, more names, more names. Centromere is the part where the chromosomes cross over. So you've seen this. Your chromosomes look really weird. They look like this. So this here be your centromere. And you know that they're kind of held together by cohesin all around. And the chiasmata is the point at which it crosses over. So recombination happens at the chiasmata. It's just like a location. Um, do we have any idea what the microtubules are doing here? Why are we saying there's microtubules here? What are they doing? I'm not getting very many answers. So remember, do you guys recognize, like, if I were to draw this in the context of like cell cycle, would you know what I'm referring to? Yes, yes. Okay, now I'm getting good answers. Yes, it is. So the spindle is made out of microtubules. And so these is that. You're pulling them apart during cell division. Yes, good. And then well, this is the spindle. And so it's doing its thing. Okay, cool. Um, so SPO11, like we said, SPO11, uh, eraser, creates those breaks. And then so SPO11 creates those breaks, right? Recombinases, they go ahead and help make it happen. Right. So you have to break something. Remember, I selected my eraser, I broke that, but then I had to draw it in again. I had to do the exchange and I had to draw it in. Recombinations are what do that drawing in. They recombine, like they facilitate recombination. They recombine the strands, make them whole again, just with different components. We cool with that? So clue is in the name. I think that's a huge kind of theme in BIOL005A is clue is in the name. Clue is in the name. You are recombining the chromosomes after breaking. Okay, cool. Oh, this is in Dr. Wells's words, not that deep. So think of this as a ruler. Okay, my wonderful fancy, my work of art that is my ruler. Okay, and on this ruler, I'm going to put down two marks. I put one on the end and the other on the end, right? And then I get really mad because it's Sunday before finals week, and so I'm gonna break this ruler in half. What is the chance that these two marks end up on the same half of the broken ruler? What is that chance? It's not 50. If I could, if it's like if I could have made, um, it's well, that's I see why I see why you're saying that. It's if I could have made this break anywhere versus like yeah, exactly. It's very unlikely that they're gonna end up. I don't know if it's exactly 50-50. It's more like this means something else. So you see how these two distances are the same and they're both 50. So the 50 means something else. Yes. So it's very unlikely. But if I were to break this ruler and I had drawn two marks right next to each other and then I broke the ruler, well, what are the chances there? Are they better or worse that I find them on the same spot? Yes. Yes, yes. I'm seeing understanding in the chat. Very good. It's much more likely that when I break the surface they're on, the closer together they are, they're going to stay together. And that's important when I take this piece of ruler and I put it on another broken piece. And I kind of Frankenstein a ruler out here. These are going to travel together. This piece, these two, they're so close together, they're not going to be separated. That's exactly what happens here. These two right here, right? If I break the chromosome like that, these two are going to be inherited together much more likely than A and E, right? They're much closer together, and it's much more likely that they'll be together. The way that I was taught it is these numbers are either chance of being separated. So the smaller you get, the higher the chance of not being separated. So smaller the chance of being separated. Um, and then also that's the length of the chromosome. So if you're 10% of the length of the chromosome away from each other, so like if we use the ruler, I'd like the ruler for this one. And on the ruler, we're like, let's say it's a 10-inch ruler. On the ruler, we're only 1 inch away from each other. There's a 10% chance that we will get separated. Whereas if we're 5 inches away from each other, the same, like the whole length of the ruler, there's a much bigger chance we're going to get separated. It's almost no chance that we're going to be inherited together. Does that kind of make sense? It's like the closer they are, the more they'll avoid the breaks and they'll stay together despite being broken. That's really poetic. But does that make sense? Look at the distance on these and where they ended up versus the distance on these and where they ended up. And so what does this mean? This means that the closer genes are together, the more likely they'd be together and crossing over. Thank you. Um, And then this was I don't think she's gonna ask you about this, to be completely honest with you. I don't know if she told you she wasn't gonna ask you about this, but whatever. It was kind of like the last 10 minutes of class. Um, okay, I see the question. It's saying that if I were to take this whole thing as, say, a chromosome, right? And well, realistically, a chromatid, and I were to do crossing over, so I would break it somewhere, like either here or here or here. I would break it somewhere. The chance of A and B being separated, so separation, that I can spell separate. That's not how you spell that. Whatever, is much less than the chance of A and E being separated. Does that kind of make sense? It's like the closer they are, whenever I break it, they're probably going to be together if they're closer. And we're talking about like A and B. So we're linking individual genes. We're not saying like A, B, and C, D. We're linking like A and B or A and D, A and C. And distance is linear. It's pretty much exactly as to how likely they are to be separated. Um, yes, yes, we can. So finish up for this, right? We can do that. We can do that. We can do that. This is setting you up for Bio 5C, by the way. And yeah, we could do this because there's only two. Loki. Nah, four. Counting the ones holding everything together. Does this kind of make sense? Does this make a little bit more sense than before? Okay. I'm seeing some approval in the chat. Okay, cool. Good. Um, let's uh we can do another unit like question in a little bit. Let's actually switch gears for a second. Let's go look at the practice final. Here's the question. Do you want to look at a practice midterm or a practice final? If so, which midterm or the final? We can look at midterm one, midterm two, or the final. Vote now on your phones. Okay, I really don't know what I was expecting, but okay. Um, cool. Let's go look at the practice final. So, did I? Oh no, go away. I'm busy. Who dares to call me right now? Okay. I'm guessing you guys looked at it. So, do we have any specific questions about any specific numbers? Or if you don't, I could roll a random number and we'll go off that random number. But I do prefer to be directed. So if you guys have questions about specific numbers, I'll go ahead and look that up. I got an okay, cool. Yeah. I didn't get a number, but I got a topic. So let's go ahead and do that. Let's see if we can find that topic. Okay. Yes. So let's go ahead and focus on this topic real quick. Throwback. Okay. I see acid-base chemistry, cycles, transcription and translation. Sure. Um, really, what would be really helpful? Oh god, there's someone in the waiting room. What would be really helpful is if you guys opened it on your own and like found numbers that we could go to. I could look around for these like ideas of these questions, but if you have a number, it'll be that much faster. So find your numbers. Okay, so this one, real quick, throwback to unit one. Let's go ahead and draw these out. So A is a COOH is a what? What do we call this one? What's the name? Well, we call it coupe. But aside from calling it coupe, what's the name? Yes. Yes. Very good. Do you guys by any chance know the full name of a carboxyl? Because carboxyl is a nickname. Do we know the full name of a carboxyl? Yes. 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 You guys are lighting up my DMs. I want you to have audacity and put it in the chat for everyone to see. Because I know you got that shit, but everyone else needs to know. Somebody put it in everyone, please. Yes, thank you. Carboxylic acid. This here be an acid. I was doing, do you guys know like that TikTok thing where it's like you point like with the pointer and it's like they're tapping with the pointer? I was trying to do that. Wait, can I do that? Do I have a thing? Oh, I don't. Oh well. Anyway, yes, so that doesn't fit the description. Uh what about an OH? What does this kind of look like, y'all? It looks very similar to something we're all very familiar with. Is somebody going to have the audacity to suggest an answer? That's the name of it. That's what it's called. Uh, yeah, okay, I'll take it. But that's what it's called. But like, what does this look similar to? Does it perhaps look similar to this? Does it perhaps look similar to this? Yes, agua. So, is water acidic? Is water a base? Is it either of those things to you at body pH in neutral conditions? I'm staring down my chat. I'm watching you guys. Okay, I'm hearing water could be acid or base. You're right, which is why I specified to you. So when you drink water, are you drinking acid or are you drinking base? Like, are you drinking something that is inherently acidic or basic? Neither is an answer, guys. And that would actually be the correct answer. Water is neutral. Yes, water is neutral. Water's pH is about 7, which is in the middle of 0 to 14. So it's about neutral. Water is neutral, therefore, hydroxyl is neutral because hydroxyl looks exactly like water. And you will use this in OChem, actually. You'll use that knowledge in OChem. The specific funny number for water that determines its acidity is 16. The specific funny number that determines acidity for OH is 16 to 19. So they are the same. They're not acid or base. So it's not this one. And we know it's not this one because it's acid. What about NH2? NH2 looks like this. Yes, it is. What does NH2 have that none of these other things have? What's unique about NH2? Yes. Okay, very good. It's the lone pair. So in chemistry, this right here means very, very negative. Well, partial negative, but like very partial negative. And acid, as we recall, is just protons. Ugh, please. Acid is just protons. It's just positive hydrogens. Positive is going to want to go to negative, which is why nitrogen can just steal a proton from the water and become what? What does stealing a proton from the water mean? What does that function? What things do that? Yes, exactly. Full circle. Bases do that. A base will steal a proton, an acid will throw away a proton. Cool. So it's amino. Um, are we familiar with base or positive, acid or negative? Do we understand why? Okay. When I have, I'll do this real quick and then we'll move on. When I have an amino, this is neutral. When it picks up a positive thing, it becomes what? Logically, if you pick up something positive, you become. Yes, positive. So a little bit of overthinking happened there, but that's okay. You become positive. So we call this NH. Well, if this is an amino, then this would be NH3 plus, and this is just NH2. So bases can become positive because they pick up positive protons. On the other hand, if an acid can let go of a proton, it will become negative because it has lost something positive. Right? So if you lose something positive, you become negative. If you gain something positive, you become positive. That positive thing in question is a proton, hydrogen ion, acid. Acids will lose protons and become negative. Bases will gain protons and become positive. Sounds good. Okay, I'm gonna assume that sounds good. Um, I'm a chem major, so I can talk about chem all day, but we're not going to do that. Does anybody have a number for me? Does anybody is 30? Oh, damn, you were ready for that shit. Damn. Okay. Let's go ahead and get a Q going. Let's start with 17. I'm going to write down this Q on my phone. So once you put in a number, there's no going back. If I can open my notes app. Uh, Q 17. Okay, cool. 17 first. Ah, yes. Does anybody remember this word? In your honest opinion, does anybody remember that word being mentioned in lecture? Cool, because I don't. I do not remember that word being mentioned in lecture. um, I remember that word being mentioned in AP Bio. That's the last time I heard of it. Definitely not in lecture. It might have been in a pre-lecture, but I don't watch those because I'm a bad SI leader. So I don't watch your pre-lectures. It might have been in your pre-lectures. Anyway, think of epistasis as a epidemic. Uh, she said it's not in the exam. Oh, I guess if you haven't covered it, okay, okay, cool. Then we'll spend like we'll spend no more than like 60 seconds on this. Epistasis, think of it as an epidemic, right? Epidemic things spread. Ooh, things spread around. So epistasis is literally something spreading, right? In terms of genes. So in this case, our answer is B. It's something that affects something else at a different location, like an epidemic. An epidemic will affect people all over the place. Epistasis is an example of a gene affecting another gene. Fair enough. We didn't really get to it this quarter, so I'm not going to focus on it that much, but think of it as epidemic. Um, oh no, I dropped my Apple Pencil. Whatever will I do? Okay, next number. Next number is 14. Okay, cool. So let's talk about crossing over. We just spent all that time talking about crossing over. Does crossing over increase the total number of potential gene combinations in the resulting gametes? So, total number of potential gene combinations. This is the smart way of saying biodiversity. Does crossing over increase biodiversity? Does it increase like differences in genes? Yes, I'm seeing a lot of yeses. Is SPO11 required for crossing over to occur? Yes, we just went over this. So, if I were taking this test, I would low key stop there because we already have two correct answers. I don't know why I drew crosses there. That was kind of weird. We already have two correct answers. It has to be D. Um, Low key, I would stop there because I don't know if it occur. Like I don't know the differences between meiosis. I'm a second year and I still don't know. And you might forget as soon as you're out of this class. But yes, absolutely. SPO11 is required. It increases biodiversity, and apparently it occurs during meiosis one. So we're gonna cover a few things. We're gonna cover a few topics. So not topics, but like tips. Let's call them tips. Identify topic. Okay. Identifying a topic is important. If I'm answering a question about um, crossing over, I don't want to be thinking about photosynthesis. I don't want to be overthinking a question about photosynthesis from 3 questions ago. I don't want to be like cluttering my mind up with all this different stuff. I want to know exactly the topic I'm talking about. After that, I want to focus on that and I want to start eliminate. Process of elimination is your friend. We really like the process of elimination here because sometimes Dr. Wells speaks College Board and there's more than one good sounding answer. Elimination will help you get down to that. And then justify. If it makes sense to you, it makes sense to her. If it sounds right, it is right. Don't doubt yourself because that's what's going to lose you points. You will know what you're talking about, but you will doubt yourself. I'm speaking from experience. So, identify the topic, do your process of elimination, and justify. So, yes, that one was D. Are we good on this question? I'm guessing we're good on this question because I'm not getting any rampant. Yes, okay. 13. Why is myosin important to the cell cycle? Myosin, talk about myosin. What is myosin part of? What topic is myosin part of? Do we know?
No,
It's okay. Yes, I got one. I got one answer. I got another kind of answer. Um, yes, myosin has a motor domain. Myosin is part of the same topic as microtubules, and somebody got it right. Myosin is part of the cytoskeleton. So in this question, we are talking about cytoskeleton and cell cycle. So cytoskeleton, is this relevant? Okay, D is not really relevant. That D is a bunch of things, but it is definitely not myosin, right? It's not part of the cytoskeleton. What about this? Does this have anything to do with the cytoskeleton? No. C doesn't really have anything to do with the cytoskeleton. Yes, those are histones. Very good. This one's a little bit rougher, but does this have anything to do with the cytoskeleton? No. Those are Well, this is kind of actually hard to answer. It's the wording of this question is really weird. It's either centromere or cohesin. It's one of those two. In any case, it's not part of the cytoskeleton. So the answer is no. We were kind of jumping around, so we did review of a unit. So we did review of crossing over and answer questions. Right now, we're going over the practice exam. We're going to keep doing this for a little bit, and then we're going to see if we have any unit related questions, and then we'll kind of like jump around wherever we need to. What is the point of the cytoskeleton? I'm glad you asked. Let's talk about let's invoke protein synthesis. Okay, you have DNA. DNA gets read. It gets written down. Call this transcription, right? I'm gonna. That's a pen, guys. That's a pen, guys. It gets written down. It gets transcribed. It gets moved to what to be translated? It gets moved to the ribosome. Exactly. Yeah, the small sort large subunit of the ribosome. Yes. It Here's why the cytoskeleton is important. It doesn't just float there. Like it does the mRNA doesn't just end up at the ribosome. Yes. I like that. I like that term. The cytoskeleton is a highway. So this is a cell. Um, the cell's name is Bob. Hi, Bob. Bob has a skeleton, a highway where things get moved around and where things sit on, and everything is connected. Right? It's a highway for transportation. It's there for structure. It's there for uh, signal communication. It's there for everything. The cytoskeleton is kind of the invisible friend that cells have. It's just it's order, it's structure, it's transport, it's everything. Um, I got asked a question. The question was the other molecule that formed the contractile ring. Yes. So myosin is most commonly found with what other protein? Yes. Very good. Myosin is besties with actin. And so actin and myosin form the contractile ring. They also form contractile assemblies. So your muscles will have actin and myosin in them. They're besties. Are we okay with number 13? Are we good with these are not relevant, so it must be A? And we know it's not relevant because we know we're talking about the cell cycle. So I'm so sorry, the cell cytoskeleton. There we go. Oh my God. And we're talking about the cytoskeleton, and we know that's not relevant. Cool. Um, let's move on to the next question. Hi, Unit 2. I'm sure you guys love Unit 2. I'm sure it's your favorite unit. Anyway, so which following the molecules is likely to be oxidized? Let's think about what does oxidation mean? What happens when something gets oxidized? Somebody. I like that. Something loses electrons. Okay. What it loses electrons, or does anybody know like the shortcut to oxidation? Would it be? I'll try to get a YouTube link. How would I do that? I'll try to get a YouTube link going. Yeah, you guys have my email, so I'll figure it out. Don't worry. Yes, Leo says grr. Very good. The shortcut to oxidation. Put my YouTube in chat. No, it's gonna be my school account. It's here. Okay, you got me distracted now, but I should do this while you guys are still here. Um, I'm seeing Leo goes grr, and I'm seeing a bunch of like loses electrons. Polar and non-polar relates to how likely it will be to oxidize. Yeah, sure. I like what Okay, and then we have some answers as well. What does oxidation sound like? And I'm getting my YouTube account for you. Shameless plug. No way, it's just called that. Okay, guys, check the YouTube. Phosphorylation, oxygen, oxygen, oxygen. Oxidation sounds like oxygen, right? Fair enough. Here's the shortcut. Get a little chem inside knowledge. Oxidation is roughly equivalent, in most cases, relevant to you, attaching an oxygen to something. Okay? You attach an oxygen to something, 90% of the time you're oxidizing it. When you remove an oxygen and attach a hydrogen in its place, what do you think happens? Reduction, exactly. So in chemistry, adding hydrogens is a way to reduce things, or like it's associated with reducing things. So we want to find one of the volume molecules are likely to be oxidized, likely to receive an oxygen. CO2, she built like this. Is she going to get any more oxygen? Is she going to be oxidized anymore? No, she done. She wide. I like that. She done. She done. She's hella oxidized. This is the most oxidized a carbon can be. What about pure oxygen? I like that. They're already a couple. Yes. So true. They are already a couple. Oxygen has enough oxygen to oxygen. It is not a triple bond. Um, I could explain why it's not a triple bond, but that's not necessarily time wise. Yeah, exactly. Yes. Um, NAD plus. Well, NAD plus can gain an electron and turn into NADH. Wait a minute. We added a hydrogen. We gained an electron. Is that oxidation? So I should say plus hydrogen plus electron. Is that oxidation? No, it's reduction. So it's not this one either. So it's got to be this one. And that actually checks out because adding an oxygen, that's way too many carbons. Guys, I'm good at chemistry. Adding a oxygen anywhere here would be extremely oxidative, right? So that's the answer. Are we cool with that? Is that sane? Does that make sense? Okay, very good. Um, let's go ahead and pause for a second. Do we want to continue on the practice final or do we have a specific topic question? Vote now on your phones. I'm watching. Okay, I'm getting a lot of. Oh, good God. Okay. Will the activity from Friday be on the exam? Um, Plan for it. I don't know necessarily what kind of questions she can ask, but do plan for it. Okay. By incredibly popular demand, let's talk about Unit 2. It doesn't matter, Julie. Whatever you want. Um, by incredibly popular demand, let's talk about Unit 2. Let's talk about photosynthesis, cellular respiration, all that jazz. I'm going to do it right here on the blank slide. So, everybody's favorite unit, right? What do you all need to start with? What does cellular respiration start with? Right? What does every living thing need to consume to live? Hell yeah, glucose. And it all starts with glucose. Uh, yes, actually, you are more correct than you think. Carbohydrates would be a better thing because you can't necessarily consume glucose. Um, we don't really consume glucose; we turn it into glucose. But yes, glucose is where it starts. Glucose is associated with a process called glycolysis. Very good. Glycolysis. What do we need to know about glycolysis? Well, I would like you to know in, out, where, and why. So, into glycolysis, we put what? We have to invest something, correct? In the investment phase, we have to invest something in glycolysis. What is this investment? I see carbon and glucose. Yes, carbon comes from glucose. Yes, so this is invested, technically. True. ATP is also invested. NAD plus is also invested. And a lone phosphate is invested. And I guess ADP, sure. Out is ATP, right? We can link this directly. Let me do it this way, and I'll show you why. You can link it directly. NAD plus, and well, let's say glucose. This turns into NADH. The glucose turns into what does the glucose turn into? Is not quite, but yes, James. Um, real quick detour. Light energy is notated as this. NAD plus is like a battery. These are your like double A batteries. It'll make a lot more sense in a second, but think of NAD plus as your batteries. Pyruvate. Yes, very good. Do you see how there's a direct link between input and output? If you are sitting on a test and you are not sure, justify it. Logic that shit out. Okay. If there's an input of some specific like flavor, there's going to be an output of that exact flavor. Right. So if there's ADP and ATP as an input, you're going to output ATP. NAD plus as an input, you're going to output NADH. Glucose, a carbon based molecule, just sugar, will turn into another carbon based molecule, pyruvate. Okay. Do we know where glycolysis happens? The okay. Yes, cytoplasm. Yes, you're right. So cyto, little factoid here. Cyto means cell. Cytoplasm means cell juice. This is happening in the cell juice. Okay. And why? No, you're that's so real. Why is this happening in cell juice? Why do we need glycolysis? Energy. Yeah, exactly. Energy intake. Yes, yes. So one of the main reasons glycolysis happens is to make this ATP. And that's exactly why it happens in prokaryotes, right? Because it's the only way they make ATP. But that's not your main reason to make glycolysis. That's not your main reason to do it. Your main reason is pyruvate. Because pyruvate, as somebody mentioned, actually a few people mentioned, pyruvate goes into the next cycle called, well, not cycle, next process called pyruvate oxidation. What do you suppose is an input for pyruvate oxidation? I see why you're saying oxygen. Indirectly. Indirectly, it's an input. It's an input from within pyruvate. Pyruvate has oxygen in it. But yes, absolutely, pyruvate. You guys are so good at this. In goes pyruvate, and out comes what? Acetyl CoA. Very good. Acetyl CoA and anybody else? One more thing. CO2. Very good. This is you breathing out. This is where humans breathe out partially. There's another way they breathe out, but we'll get to that. And of course, you have your batteries powering all of it, right? You constantly have these batteries, your little Duracells, your little double A's, powering everything. They're powering this, they're powering that. They're not used anywhere else. They're used to power the cycles. Okay. What about after that? Well, you've just made acetyl CoA. You got to do something with that acetyl CoA. It's got to be an input somewhere else. Where is acetyl CoA an input? The little batteries are NAD plus. So your high energy electron carriers, I'm referring to as batteries, and it is because they can carry electrons, they can carry electricity, literally, and like dispense electricity, pick it up, be recharged. They're like batteries in every way. Yes, FAD plus, FADH2 are also batteries. NADB plus and NADPH are also batteries. So like that, I like to think of them as batteries because they literally carry electricity. Um, I'm seeing citric acid cycle, Krebs cycle, fermentation or Krebs cycle. Very good. The Krebs cycle, TCA or the Krebs cycle or the citric acid cycle. Let's talk about the citric acid cycle. This was kind of cool. Acetyl CoA, a two carbon sugar. We know this because a three carbon sugar lost CO2. So now we have a two carbon sugar. A two carbon sugar will join with a four carbon sugar to form, let's do the math. Ooh, okay. You're jumping the gun there. Damn, you're stealing my thunder. Hell yeah, you got it. And Sergio stole my thunder there. It's called what? It's called citrate, as in the citric acid cycle. So the citric acid cycle, the first step in citric acid cycle is making citric acid. The first step in pyruvate oxidation is oxidizing pyruvate. The first step in glycolysis is lysing glucose because lysing means to split. Think about hydrolysis. So these processes are named for what they do. Glycolysis, splitting glucose. Pyruvate oxidation, oxidizing that pyruvate. And the citric acid cycle immediately makes citric acid. But moving on, you start to bleed carbons. Okay, and you start to bleed carbons. Where do you suppose these carbons are going? Breathing out. Yes, I like that. Breathing out. CO2. This is where your CO2 is generated and you breathe them out. Very good. Now, this might be like a ghost zone for some of you. The good news is you don't need to know that. Because guess what isn't on this final? For all the things that are on this final, there's something that isn't on this final, and it is an FRQ. There is not one FRQ on this final. Your ass is not drawing anything. So take some solace in that. Unless you're good at FRQs. In which case, I feel you. OChem is all FRQs, so you'll get your chance. But your ass will not have to draw anything. So you don't need to know this. But for your enrichment, because you guys should be enriched, there are four stages of four quadrature. And that's funny because there's four fours. Anyway, that's not the important part. We still have our batteries. Our batteries are still doing the work every time, right? Do you see now why I call them batteries? They're out there powering literally everything. Okay? Now, coming down, coming down from this kind of fall from power from six carbon to four carbon, there's a lot of energy going on. This energy is going to be used to sneak an ATP. Just a little thing on the side, little side hustle. Okay, the citric acid cycle is not there to make ATP, but it's a little side hustle. It's there to just do this thing, right? You okay with that? Yes. And side hustle ATP, we've already talked about. This is side hustle ATP. ATP. Yes, exactly, Sophie. So this is a side hustle. This is a side hustle. That's not their main thing, right? So it's something, you know, under the hood. It's something done on the side. It's something done at a substrate level, literally a lower level. That's when you turn ADP into ATP as a side hustle. That's what substrate level phosphorylation is. And that happens in glycolysis, that happens in the citric acid cycle, right? Is that coming together a little bit? I'm going to trust that that's coming together. Anyway, all that jazz doesn't matter. The main part of this is to generate batteries. Actually, the main point of the citric acid cycle is to make NADH and FADH2. Now, hey, y'all, I just made NADH and made FADH2. I made NADH. I made NADH. I got a whole bunch of this stuff now. Where am I going to go ahead and put that? Yes, electron transport chain. I've just used all these processes, and theoretically, actually, you could say that the purpose of all these processes is to charge those batteries. I've sat here and I've charged my batteries. I've got a bunch of NADH and FADH2, right? Now I'm going to use them. Now I'm going to go ahead and draw this wonderful mitochondrial electron transport chain. I'm an artist, if I do say so myself. I'm going to donate my NADH and bring back my broke ass NAD plus. I'm going to donate FADH2 and bring back my broke ass FAD plus to get put back in the cycle. And these are just going to move an electron along, right? Um, how are we on understanding the electron transport chain? Remember, there's no FRQ, so you don't need to know it piece by piece, right? I know she had you guys draw out like a process on the midterm. You don't need to do that here. You don't need to know it piece by piece. You just need to know what it does. And what it does is transport electrons exactly to form an H plus gradient, right? From these pumps, you're pumping in a gradient. Okay. Where does oxygen come from when it accepts electrons? Well, um, remember your cell wall. Remember, oh, not cell wall, cell membrane. Remember what can pass through your cell membrane. CO2 can pass. O2 can pass. H2O cannot pass. Glucose cannot pass because these are polar, right? And this is polar. They can't just pass through unassisted. That's fire. This can pass through though, so your cell constantly have has a bunch of oxygen in it, right? So it just comes from around. It just comes from around. It's just there. But this oxygen is involved here. For the first time, you're involving oxygen and you're making ATP. This is called oxidative level phosphorylation because you're involving oxygen. This is the only place your body uses oxygen. That's not true. This is the only place that you know of that your body uses oxygen. And it's actually really cool because if you were to replace this oxygen with a cyanide, it takes it. Complex 4, this little dumbass over here, recognizes cyanide as oxygen and takes it. And it stays there. And it doesn't leave. And the cell shuts down. And that is why you should breathe oxygen and maybe consider not taking cyanide. So this is bad. This is good. Glad we got that settled. Okay. So what's important for the electron transport chains? Important for the electron transport chain is a chain of events. You donate, so NADH, FADH2. You make a gradient, you make ATP. And you accept at the end of this gradient with O2. In photosynthesis, it's exactly the same. Is that why cyanide kills? Absolutely. That is exactly why cyanide kills. Cyanide binds to complex 4 in your mitochondria and says, no more for you. And it shuts down the circuit. In photosynthesis, the electron transport chain, you have to have a donor. Does anybody remember the donor? It's a little bit different, but it's not much different. You'll see the important part is the same. So you'll see it in a second. Does anybody remember the donor for photosynthesis? Not quite. I have one right answer. Let's think about this on a bigger scale. I always like to have you guys zoom out. What am I drawing here right now? Oh shit. I fucked that one up. You didn't see that? What did I just draw here? This equation right here. You guys are all familiar with it. Not quite cellular respiration. Inputs and outputs, sure. But think of it as an equation. Think of this as an equation. Water plus CO2 goes to glucose plus O2 using light. The plant thing, yes. What is the plant thing called? What is the thing that plants do called? Hell yeah, photosynthesis. And so water gets used as the donor in the electron transport chain. CO2 gets used in the, so this is the electron transport chain. CO2 gets used in the Calvin cycle. Light gets used in the electron transport chain. Glucose gets made, right? And then used, actually, it gets made in the Calvin cycle. And O2 gets made in the electron transport chain. Everything here checks out. Water, light, and O2 are in the electron transport chain. Carbon dioxide, glucose are in the Calvin cycle. So remember, this is literally the same thing. You're doing the exact same thing, except here the acceptor is NADP plus. because P is for photosynthesis. So I think the most important takeaway from this is the donors and acceptors, actually. So let's actually focus on that. In mitochondria, NADH and FADH2 will donate and turn into their other forms, right? So donate. Acceptor, oxygen, will turn into water. In the photosynthetic electron transport chain, the donor is water. Now this is gonna connect. Okay, this is gonna click. This is gonna be really cool. Plants have mitochondria. Plants will make water from the oxygen they breathe. Plants will use that water to donate into the electron transport chain of photosynthesis. What do you think they make? Not quite, not yet. Eventually, ATP, sure, but in terms of the donor. See how this donor turns into this. This donor turns into this. This acceptor turns into this. What does this donor turn into? One of you got it right. There you go. Oxygen. This is where plants breathe out oxygen. And they don't just breathe it out, they breathe it right back in their mitochondria. Isn't that so cool? They kind of cyclicize their cycles. That's not a word, but you know what I mean. Anyway, that's the important parts. And then the acceptor is NAD plus or some dumb shit like that. That's not as cool as oxygen and water being changed out. This is the important part of the electron transport chain. And the fact that there's a gradient going on. Are we cool with that? Let me really quickly run a relevant question to this. I think, no, actually, I think that was on the midterm. Yeah, that was on a midterm. Basically, the question was which of the following is maybe it was on the final here. Was it on the final, please? No, it was on a midterm. Damn it. The question was, let's go look at it real quick. I promise this is important. Where's your midterm? Where the hell is your midterm? There it is. This question. This is the kind of question she can ask. Hell yeah, Julian, that's correct. This is the kind of question she can ask, right? She can go, the electron transport chain of photosynthesis, the donor is, and the acceptor is something, right? This is where you would think about it. There's two ways to do it. One way is to go, okay, I know what photosynthesis is. I know that plants need water to survive. I know that plants need carbon dioxide to survive. So this is not an input. This is Brad. Because between those two, I also know that plants don't produce water, they need water. So it can't be this one. Right? That's one way of doing it. Another way of doing it, I think the faster way of doing it is saying, hey, this is an electron transport chain. What flavor of molecule, what brand of molecule do we need for an electron transport chain? Spoiler, it has the word electron in it. So the question was, what kind of molecule do we need for the electron transport chain? Yes, Julian, you said it correctly, but yes, Sergio, yes, Sergio got it. What kind of molecule is that? What's the brand? It's a kind. It's a flavor of molecule. Yep, that's correct. Is someone gonna say it in everyone chat though? You're correct, Sophia. Come on, it has the word electron in it. Powers electron transport chains. To do that, it has to carry an electron to the chains. So it is a hell yeah. So a bunch of people messaged it to me, but I wanted to be in everyone chat. You need high energy electron carriers somewhere, either an input or an output. Somewhere there's going to be a high energy electron carriers. In looking at this question, which one of these answers is the only one to have a high energy electron carrier in it? And that ends up being your correct answer. You don't even have to look at the other ones. You don't even have to think about it that much. The only electron carrier is C. It can only be C because you need electron carriers for an electron chain. Anyway, that was my like. I'm passionate about that question because I think it's really cool when you could just say, "I'm not actually reading this question. I'm just going to do that." But anyway, yeah. So summary for the cycles for Unit Two: electron transport chains. Ugh. Electron transport chains. You should know the ins and the outs. Other processes, you should kind of know in and kind of know out, like pretty well, especially for glycolysis. But mostly you should know where and why. So, not squiggly. You should mostly know where and why. That being said, I don't think she's going to ask you about fermentation. She hasn't asked it. And we covered it a lot, but she hasn't asked it. So I don't think she's going to ask you about fermentation. Still, it's best to be prepared. Um, are we okay? Oh, okay. And then also, does anybody know what's the only enzyme you guys are supposed to know? That's true, James. Yes, Sergio. Ooh, is it RUBP or is it Rubisco? Okay, it's Rubisco. Yes. So Rubisco is the only enzyme you should know. Rubisco, uh, it's the only enzyme that doesn't have ACE on it that you should know. It doesn't have ACE, but it is an enzyme. And Rubisco just does stuff with RUBP. So it works on RUBP. Right? And where does this happen? Real quick. The so true, King. the not the mitochondria. Sergio's got it. The Calvin cycle. So this happens in the Calvin cycle. Think about it this way: Rubisco has CO on the end of it. CO is there for CO2. Yes. Does the Calvin cycle happen with that? No. So a chloroplast is built like this. Do we agree? The Calvin cycle happens here. The electron transport chain happens here. Well, the Calvin cycle just happens around the I believe she's called the stroma. It happens in the cell juice inside the chloroplast. It happens everywhere. It's kind of like how glycolysis happens in the cytoplasm. The Calvin cycle happens in the cytoplasm of the chloroplast, which is called the stroma. Are we okay with that? I'm guessing we're okay with that. Let's go ahead and switch gears. First of all, did that like help at all? Are we like roughly okay with that? I know this is a lot of shit. This is the highlights. Okay? This is the highlights. For the electron transport chain, oh fuck. For the electron transport chain, your ass better know the inputs and the outputs. Because she's asked that kind of question. For the other ones, glycolysis you should know for sure. Glycolysis you should have down. But for the other ones, you should mostly know why they're there and like what they are. What happens inside a thylakoid? Well, I'm glad you asked. This is how a thylakoid is built. This here is a thylakoid. Over here in the thylakoid, we have the Calvin cycle. It just happens, right? It just happens around. Here, I'm going to use like a blue green on this membrane on the uh oh fuck. I lied to you. This shit is not a thylakoid. My bad. This is a chloroplast. This is a chloroplast. That is a thylakoid. Okay, cool. On the thylakoid membrane, so kind of like the inner membrane of the mitochondria, you have the cycles going on. And here's a zoomed in version of that. You have a PlayStation, PlayStation 2. You have another PlayStation, PlayStation 1. You have ATP synthase. And then you have a few pumps here and then a bunch of kind of irrelevant proteins. Okay? The big picture with this is that in photosystem 2, which is this thing right here, you receive electrons through water. This happens here. Okay? Um, and then these electrons, I'm going to draw the electron in orange because orange is kind of electricity E. Actually, no, I'm going to draw it in yellow because yellow is even more electricity E. The electron is going to travel. It's going to kind of stay here. It's going to hit this right here. That thing in the middle. That thing in the middle is a special pair of chlorophyll. It's going to sit there. That electron is going to sit there, right? While that electron sits there, light is going to come down and hit this photosystem. This is going to send energy, not electrons, energy down the photosystem. That energy is going to hit that electron, and that electron is going to go fucking flying. And in the like time that it goes flying, because it just got hit with a bunch of energy, is going to power everything on the way, right? These are pumps. And these pumps are going to be powered by this electron that's currently flying because it got hit by energy. This electron is then going to sit here. It's going to sit at a different pair. Same idea, just a different pair. Like a bad cameo, light comes back and is going to hit this photosystem right here. Say it with me. The energy is going to go down and is going to hit the electron. And this electron is going to get launched again. Except this time it's kind of bad because it's not accurate. So it's either going to get launched back here, where it just repeats the process, or more likely, it just gets launched along, where it just completes the chain and gets picked up by an ADP plus. That is what happens on a thylakoid membrane. That's the only relevant part that happens on a thylakoid membrane. Everything else is kind of irrelevant. Well, I mean, there's ATP synthase too, but same thing as mitochondria. Okay, cool. Let's go ahead and switch gears. Do we want to continue with the test and we have any numbers to answer, or would we like to cover another topic? Okay. Cell cycling question 35. Let's do question 35 real quick and then we'll do the cell cycle. Actually, yeah, this one's a pretty quick one. But then we'll do the cell cycle. Um so question 35. Membranes of a eukaryotic cell. What are membranes made out of? What do we call them? They're made of lipids. What kind of lipids? Phospholipids, right? Membranes, all cell membrane and cholesterol, you're right, and some fatty acids. But all cell membranes are phospholipid bilayers, right? They're all phospholipid membranes. So can A be true? In what way do membranes vary? How are they different? Is A true? So if all membranes are made of phospholipids, can it be true that phospholipids are only found in certain membranes? Yes, exactly. It's not true. Every membrane is made out of phospholipids. What about only certain membranes are selectively permeable? Is that true? That one's a little harder. So let's leave that for a second. Let's look at D. Only certain membranes are constructed of amphipathic molecules. She's throwing a big word at you to scare you. Okay? Be not afraid. Your ass better be not afraid. Because amphipathic just means what? You're halfway there, Sergio. Yes. It is both. It is hydrophobic and hydrophilic. So think of amphibians. Amphibians exist in both land and water. Think about people who are ambidextrous. Think about ambiguity, right? It just means both, either or both, right? And so amphipathic molecules are both. Only certain membranes are constructed of amphipathic molecules. No, the entire point of a membrane is to be amphipathic, all right? So this can't be true either. We're down to B and C. Does anybody have a guess for me? We're thinking B. Are we okay with that? If you're unsure, go C. Let's talk about this. Let's talk about this. Certain proteins are unique to each membrane, or certain membranes are selectively permeable. Well, I'll have you know, all membranes are selectively permeable. Membranes are built to keep things out, but not everything. Like, for example, every membrane you know of somehow pushes um, oxygen in. Oxygen is present in every membrane, across every membrane. And if they're completely impermeable, then oxygen can't get past. So they all have to be selectively permeable, right? All of them let some things through, but not everything. So it has to be B. And B is correct. And B is correct. Here's an example. Where can I find nucleoporin? What membrane can I find nucleoporin in? The nucleus, right? The nuclear envelope, the nuclear membrane. Where can I find aquaporin in? It was a wall. I'm going to take that to mean cell membrane. So, yes, they're in cell membranes. What about ATP synthase? That's a protein. That's a big protein, but that's a protein. Can I find ATP synthase on a nuclear envelope? Can I find it on a cell wall? No. I can only find it in the inner membranes of a few organelles, like mitochondria and chloroplast. So B is true. Certain proteins are unique to each membrane. Okay? Okay, cool. I got a request for cell cycle. I'm gonna do my best because I kind of don't know the cell cycle because I never really cared and I didn't really need to because it wasn't a huge part of it, but still look at it. Um, are you talking about the cell cycle, like the stages of the cell cycle, or are you talking about like the cell cycle activity? Mm, okay, I got you for the activity. The activities, I got you. That's actually interesting. Let's pull that up. Uh, and just for warning y'all, technically this thing ends at 8, but I'm gonna stay a little longer to answer questions because I know y'all got that shit tomorrow, and I do not envy you. So I'm gonna stay a little longer. But feel free to be dismissed whenever you want. I mean, not like you need to be dismissed. Anyway, oh shit. Are we good on number one? Why might it need to divide? Well, I mean, why does cell division happen? Well, prevent. Yeah, okay. I'm seeing heal wounds and prevent mutation. I'm seeing growth. So need a cell to divide, sure. Heal wounds, grow. What about reproduce? Well, when you want to reproduce, you want a cell to divide, right? And then why might a cell need to not divide? Well, if it's damaged. And if the instructions, I like talk about an IKEA table. Like if you have a table from IKEA and you have your instruction sheet, if you break like a leg of the table, you could just rebuild that leg. Like it's not that bad. You just reattach it, right? That's like physical damage. That's what happens when you have wounds. Like there's physical damage, you can fix it. It's fine. Your instructions are still there. But if you like rip apart, if you shred the instruction sheet from IKEA, then you're in trouble because then you're going to build that table wrong. That's DNA damage. DNA are your instructions. That's like radiation or chemical or kind of mutagenic damage is when you like destroy the instructions you used to build that table. You can't really do much. You either build the table on a guess, which, biology isn't exactly building tables, it's a little bit more complicated. So that's a really bad guess. Or you kill it and you destroy that damage, which is why cells will not divide. Cool. Um, this one right here. This is just like, you know, ye old helicase, DNA polymerase, topoisomerase. That's an M. Isomerase, ligase, etc., etc. Yes, Julian. Apoptosis is a mechanism by which the body, your brain, right? So I'm going to go ahead and draw a brain. It's a really bad brain, but you know what I mean, right? And then your brain, it sees a cell, and the cell is like baked, like it's damaged, and then it basically goes, and the cell goes, and then it explodes, and so that's apoptosis, and that's to prevent damage spreading. Um, what's topoisomerase? And yeah, okay, you're good. Topoisomerase. So DNA starts in a helix. Familiar? DNA looks like this. do we need to know necrosis really? If you're going into medicine and or other health related fields, eventually, not for this class. Not for this class. Um, but if you want to work on it, you have to unwind that DNA, right? You have to do that. Topoisomerase does that. Helicase does this. It breaks the hydrogen bonds. It unzips the DNA. Um, and the single-stranded binding protein, that's just when you have your DNA and it's open and you're working on it, these single-stranded binding proteins are there to bind single strands, make sure they don't mix, make sure they don't get tangled up, right? And they just keep them like, yes, prevents reannealing. It just keeps them separate. It binds the single strands together. Um, let's talk about question three, though. Describe the process by which these proteins are made, including all necessary proteins. I mean, how do you make proteins, y'all? It's transcription and translation, right? Transcription, you take DNA, right? You find a you find a promoter, you find a coding sequence, you put a transcription factor on that promoter, RNA polymerase starts to read. I'm sorry, me when I lie. You put a transcription factor on an enhancer, the RNA polymerase goes on the promoter, RNA polymerase starts to read, and as RNA polymerase reads, it prints a copy. This copy is called mRNA. This mRNA is then taken to a ribosome and it is processed, and mRNA is printed into protein. And tRNA is the busboy that keeps bringing in resources for that protein. That's like transcription translation in 20 seconds or less. And then you have your A, P, and E complexes. I guess that might be important. But function is more important than details to her. Does that kind of make sense? So that's the answer to number three. It's basically saying describe transcription translation. Are we cool with that? Are we familiar with transcription translation? Okay, cool. How does gamma, that letter's fun to write, radiation affect the frequency of cell division? Well, if you have a cell and that cell is dividing at some rate, and then you break the instructions for that rate, do you want it to speed up that division or slow down that division? You break the instructions. Do you keep building the table if it's like 1,000 leg table, or should you like slow down? You slow down. So it slows down quite a lot, almost entirely. And the answer to this question kind of leads into it. Don't spread damage. That's bad. And then was the question more about like all these proteins, all these like weird names? P53. Okay. When let's speed run this. Speed run this in the last minute. While gamma radiation, right, it hits the cell. The cell receives stress. Stress signals P53 to be made. P53 here is a protein, specifically a transcription factor. This P53 will go and find the gene that makes P21, right? And it's going to sit on it, and then P21 is going to be made. Once you make P21, I'm going to move over here. Once you make P21, P21 is going to find a cyclin plus a CDK. It's going to combine into a megazord with them. It's going to do a 3-unit complex, so 3-member complex. And then this is going to change or activate a little guy called RB. RB is going to shut down the genes that make DNA polymerase, ligase, helicase, all that stuff, right? RB is going to talk. So I'm going to actually go down here. RB is going to go ahead and hop onto E2F. E2F is another transcription factor that helps promote making of like all these DNA proteins. RB is going to shut down E2F. It's going to stop it from acting as a transcription factor. It's something we call a co-repressor, right? And once you stop making DNA polymerase, so E2F is the transcription factor that makes like DNA polymerase. If you can't make DNA polymerase, you can't have cell division because you can't duplicate DNA. So that's how that kind of cascade works. Radiation causes cell stress. Cell stress makes P53. P53 finds P21 and expresses it. P21 combines to a megazord with these cyclins, modifies this little guy called RB. RB blocks E2F. E2F is responsible for DNA polymerase, so RB breaks DNA polymerase production. I know that's a lot of words. I don't think it's necessarily like memory worthy. I don't think you should memorize it. I mean, if you can, if it comes naturally to you, then yeah. But I don't think you should worry about memorizing it that much. It's I doubt this will be a test question saying, "Hey, draw me that." Like draw me this cascade. But yeah, this is more to teach you like the concept of a cascade. And a cascade is just like a waterfall of signals that like one thing causes another signal, which signals to another thing, which signals to another thing, which like, right? It's just that. It's just like it's teaching that one thing can cause a butterfly effect in the cell. Will there be free response in the final two? Hell no. The final is 40 MCQ. There is no FRQ. So that means no drawing. Yippee, 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 yippee. Put a heart there. Put another heart there. No drawing, no remembering, just all that shit. What topics do I suggest focusing on? While the test is cumulative, I think she said there will be more of a focus on recent top. I think she said it was some what teachers like to do is they'll do so a final. Teachers like to do final is 25% midterm one, 25% midterm two, and the other 50% is post midterm two. Um, so naturally, I would suggest focusing on the things post midterm two, but you should also focus on things that like aren't going to be FRQs. Like you don't need to know how to draw nucleotides like bonded to each other. Like you don't need to know how to do this. Like I think you should not focus on that because that's an FRQ. And I don't know. I mean, you can ask questions about like 1,4, 1,6 linkages, right? But maybe not needing to draw them. The takeaway there is focus less on brute force memorizing things like electron transport chain and more wide knowledge, less specific, more wide, and focus on things like midterm two. Also focus on things you didn't do well on. Like if I were taking this class, I would probably lock it in on like low key this stuff. Like I would lock it in on cell cycle and all that stuff because I never did well in cell cycle. Not in high school, not in college so far. So I'd lock it in on things that I don't know well. but also okay. I've been talking for the last like 5 minutes. The visuals are on the board. That's what you should focus on. And also things you're bad at. Okay. Technically it's over, so you guys are free to leave, but I will answer questions. Of course. Um, should you know photosynthesis well? Um, you should know the acceptors and donors, and you should know the rough how many extra points will discussion give you. I can't answer that any better than your syllabus. So I can't answer grading questions because I'm not in on that. I can't answer that any better than your syllabus can. But for the photosynthesis question, donors, acceptors, and uh, like know the equation. Because if you get lost, if you're like, shit, does the Calvin cycle produce or use CO2? Well, you know that plants take CO2 in, and you're like, does it use or produce water in the electron transport chain? And it's like, well, they use water and they produce oxygen and they produce glucose, right? So I would know that equation. That equation would help you out. Can I enter? Um, I mean, sure. I did. I just. I mean, sure. If you find this helpful, I can email this to you. Will I be posting on? Yeah, I'll be posting it on my YouTube channel if it loads, which I hope it does. I've never recorded on Zoom before, but here's my channel. It's my school account. So I'll put I'll post it there if it actually records because I don't know how the Zoom recording shit works. And I hope it does. But yeah, it's not like official, so I don't have like a UJ or anything for this. Unfortunately. Okay. Any other questions? Hit me with your questions. What's the YouTube link again? Oh, my fucking bad. I just sent it to one person. My fault. There you go. I DM'd it to one person. My fault. Of course. Yeah, no worries. Good luck, you guys. Um, is this recorded? I'll try. It's Zoom is recording, and I'll try to post it on that YouTube channel. But if Zoom says no, then I will be, yes.
Thank you, Ardo. Appreciate you.
Of course. Of course. Of course. Um, yeah. So let me know if you have questions. I'll be around for like, I don't know, 5 more minutes or some shit like that because the dog is hungry. So thank you. Thank you. Purpose of translation and transduction? Did you mean transcription and translation? The okay. So transcription is okay. All the instructions your body will ever have for making anything that your body makes is in your DNA. So DNA is like an IKEA manual for your body. Transcription um, is reading DNA and like it's just reading DNA. Translation is making protein based on those instructions. So transcription is reading the instructions to make a protein. Translation is actually making the protein. And then DNA replication is slightly different. There's not an explicit transcription translation. It kind of happens on the spot. It reads and makes DNA at the same time. Transcription and translation proteins are made, they're read in one place, they're read in the nucleus, and they're translated in the ribosome. So proteins are made as a group effort, but DNA like kind of keeps to itself. Of course. Thank you. Can I go over the anatomy of a nucleus? I sure can. I'm going to use this blank spot here. This here be a nucleus. This here be a cell. This here be an avocado. This nucleus has a little thing inside it called a nucleolus. The nucleolus does a bunch of stuff, mostly makes ribosomes and like it makes stuff. Inside the nucleus, this is going to be blue. Inside the nucleus is heterochromatin and euchromatin. And those are just different flavors of chromatin. Those are DNA. So remember, chromatin is just DNA wrapped around histones. This is chromatin. So there's a bunch of chromatin all around the nucleus. This is called the nuclear envelope. Right? This membrane. The nuclear membrane is called the nuclear envelope. Um, and let's zoom in, actually. Is a phospholipid, bilayer, double bilayer, whatever. Um, so a tetralayer, I guess. There are holes in the nucleus called nuclear pores. Nuclear pores are made out of proteins that sound like nuclear pore, nucleoporin. So these pores, which I would not draw in red, pores are made out of nucleoporin. Right here, right here, right here. This is nucleo. That's getting too thick for how small we are. Can I make it thinner? Yes. Nucleoporin is making up this pore. There is a component inside the nucleus. So this is inside. This is the nuclear basket. It looks like a basketball hoop. Okay. That's bad. Hold on. Looks like a basketball hoop. It's there. The nuclear basket is there to help make DNA do stuff, carry cargo in and out, a whole bunch of stuff. On the opposite end is cytoplasmic filaments. These are microfilaments attached to the nuclear pore. And they do what microfilaments do. They bring in cargo, they bring out cargo. They have importin on them, which is the protein that imports stuff into the nucleus. So they're like, if you think of the cytoskeleton, which I will draw in orange, you think of the cytoskeleton as um, all around. The I shouldn't draw it on this side. There's no cytoskeleton inside the nucleus. I should say that the nucleus does not have cytoskeleton inside of it. So more like this. If the cytoskeleton is around and it's the highway of the cell, then these cytoplasmic filaments are the on ramps to that highway. They connect the nuclear basket to the highway, right? I'm sorry, the cytoplasmic filaments. They connect the cytoplasmic filaments to the cytoskeleton, right? So this is the highway, and then this is the on ramp, and this is like the street. On the other side, same thing, on ramp, and it connects to whatever's going on in here. So this is the important part. This is the anatomy that she cares about, the nuclear pore. But this is like the other anatomy. But this is what she cares about. Does that make sense? Okay. I'll take one more question and then I gotta go feed the dog. Cytoskeleton is all over the cell. I'll take one more question after that. But cytoskeleton is so if we draw a cell, if we draw a cell and there's a nucleus and there's a rough ER and there's a smooth ER and there's a Golgi, there's ribosomes floating around, whatever, right? The cytoskeleton is everywhere except the nucleus, but it is literally everywhere. It's like it's everywhere. It connects everything. That's why you usually don't talk about it and don't draw it. It's because it literally connects everything. All of the organelles sit on the cytoskeleton. It's the fabric of reality inside the cell. The cytosol just kind of flows around the cytoskeleton. Does that make sense? It's kind of hard to wrap your head. Look at pictures. Go look at pictures of the cytoskeleton. Go look at pictures of like kinesin and stuff, and it'll be easier to understand. Okay, one last question because the dog is hungry. Of course. Good luck on your final. You're gonna do great. Or finals, plural. You'll do great. I'm guessing that's no more questions. So have a good day, y'all. I'm gonna try to get this recording up and out of the hands of Zoom. We'll see what happens. But have a good day. Good night, I guess. Have a good evening. And good luck.